master principles of inheritance and variation with high-yield NEET PYQs and exam-focused concepts in one powerful session.
Which disorder is caused by the substitution of glutamic acid by valine at the 6th position of the beta-globin chain? This is sickle cell anemia. This point mutation causes hemoglobin to polymerize under low oxygen conditions, leading to sickle-shaped red blood cells. Match list Y with list 2. Incomplete dominance is seen in flower color in antirrhinum. Codominance is in ABO blood groups. Pleiotropy relates to phenylketonuria. Polygenic inheritance is human skin color. The correct answer is A2, BIV, C3, DI. Regarding sex determination in honeybees, which statements are true? The correct statements are A, B, C, and E. Males are haploid and develop from unfertilized eggs by parthenogenesis. Females are diploid. This system is called haplodiploidy. What is the 6th mutant codon of the beta-globin gene that causes polymerization of hemoglobin? The normal codon gauge for glutamic acid is mutated to gauge for valine, resulting in sickle cell anemia. In a grasshopper species, members with 23 chromosomes and 24 chromosomes are present. Grasshoppers have an XOXX sex determination system. Males are XO with 23 chromosomes, and females are XX with 24 chromosomes. Thus, the members are males and females, respectively. In which animal do haploid cells divide mitotically to produce gametes? In male honeybees, haploid cells from unfertilized eggs divide by mitosis to produce sperm. This is an exception, as in most animals, meiosis produces haploid gametes. Select the incorrect statements regarding Rh grouping. Statements A and E are incorrect. Erythroblastosis foetalis occurs when the mother is Rh-Va and the fetus is Rh+Va. Anti-Rh antibodies are administered after the first delivery or miscarriage, not after the second child. A disease caused by an autosomal primary non-disjunction is Down syndrome, also known as trisomy 21. This results from the failure of homologous chromosomes to separate during meiosis I, leading to an extra chromosome 21. The chromosomal disorder caused by the presence of an additional copy of chromosome 21 is Down syndrome, which is trisomy 21. This leads to a total of 47 chromosomes instead of 46. In ABO blood grouping, if both A and B antigens are present, the blood group is AB. This is due to codominance, where both the I superscript A and I superscript B alleles are equally expressed. If both antigens are present in the ABO blood group system, the blood group is AB. Both I superscript A and I superscript B alleles are codominant, resulting in the expression of both A and B antigens. Identify the wrong statement about the gene I that controls ABO blood groups. Statement 1 is incorrect because I superscript A and I superscript B are codominant and produce different types of sugars, not the same. The phenomenon of pleiotropism refers to a single gene affecting multiple phenotypic expressions. For example, the HBB gene mutation causes sickle cell anemia and affects various organs like the blood, spleen, bones, and kidneys. If RRYY produces round yellow seeds and RRYY produces wrinkled green seeds, what is the phenotypic ratio of the F2 generation? Following independent assortment, the dihybrid cross results in a phenotypic ratio of 9:3, 3:1. Question 6 asks to select the correct match. Sickle cell anemia is an autosomal recessive disorder caused by a point mutation in the beta-globin gene on chromosome 11. Therefore, option 1 is the correct match. In a cross between two heterozygous individuals for the sickle cell anemia gene, the genotype ratio is 1 normal, 2 carriers, and 1 diseased. The percentage of diseased progeny, HBS, is 1 out of 4, or 25%. Klinefelter's syndrome involves 47 XXY males. They have overall masculine development, but also express some feminine characteristics like gynecomastia. Affected individuals are sterile. Thus, statements B and E are correct. A broad palm with a single transverse palmar crease or simian crease is a characteristic feature of Down syndrome, also known as trisomy 21. This condition is an autosomal aneuploidy. In human pedigree analysis, mating between relatives or consanguineous mating is represented by a double line connecting the male square and the female circle. Therefore, option 4 is the correct symbol.